Thank you.
Okay, now that Chuck is here. So I am Carmen Aguilar. I'm a scientist at the School of Freshwater Sciences, and it is my distinct pleasure to introduce today our colloquium speaker. That, sometimes they go, well, he needs no introduction, but actually, he does, everybody really knows him, and after 33 years of being on this campus, he definitely has made an imprint, and it's going to be really sad to see him leave, but he'll be back. He's going to stay away from those lumos. And the passion that he has for research, for teaching, and for this campus is definitely palpable, as we can see here. So I met Chuck ancillarily because I'm really good friends with Barb. And so, he was the husband of my best friend. And little did we know that, the path that it was going to be. At the time, I was a graduate student in Ken Nielsen's lab. That obviously he was working also with bioluminescence, but I chose the path of the trace metals and the manganese world. That is another really excellent super Shwanella world. So, I was thinking that Chuck probably would decipher what this is by now, but he hasn't said anything. So, if anybody knows, right away. So now this is the evolution of the legend that is now retiring. So then everybody really wants to talk to, take his brain, and understand some of the things that he really does. We're going to touch upon some of the things. The progression of his career, he was at UCLA as a BS in 1975, did a master's at the University of Georgia in 1978, and went back to California, to the University of California in LA, and UCLA again in 1984. Postdoctoral fellow at the Brookhaven National Laboratory from 1984 to 1986. Unfortunately, we have him now at the University of Wisconsin-Milwaukee since 1986. So he has had a great progression, and make no mistake, he didn't work on lumos all the time. He has had a really tortuous path working with plants, very tortuous path, working with plant and algal molecular biology, plant evolution, bacterial bioluminescence, and microbial ecology and evolution. And I picked a lot of these plates because, as you know, when you're working with microbiology, you have to be surrounded by plates all over. And he kind of sent me also some pictures about some of the other organisms that he worked with, some saprophytic plants, from there to Lumo. So he has really worked all throughout the plant kingdom, and with a splash of the cryptomonas. So now we know that he has had lots of different students, and we have a lot of the students here. And I want to put you guys on the spot. Would you stand up, please? The ones that are representing. We have undergraduate, graduate students, research. Being graduate students and undergraduate students. He has taught a lot of different courses in honors. He has been very prolific in things that are not only microbiology or physiology, but as you know, music is another passion. And so he has taught a lot of that a lot of different things that we have seen. He has been kind of in a lot of committees, directed a lot of theses, and colloquium coordinator at this colloquium for 17 years. So that is a really big endeavor, and we thank you for that. I know Trish does a lot of public lectures, 5 science bags, and whoever has done a science bag, you know that it's a lot of work. And you know the platform here, and numerous science fairs, and also the National Ocean Science Bowl. He participated being the person that reads those questions and maintains that dynamics in the room. So it's a privilege knowing you and being your friend, and I just know that it's a lot more years. And now Florida, yeah, of course. So more science, more Lumos, and that. So now Russell is going to say some things.
Okay, so Chucky has really integrated a love of a variety of different things, all which have some kind of science associated. Here in 1993, there was a symposium about zooplankton ecology, which you all know that Chuck is extremely proficient in. And it was run by Dr. Strickler and Dr. Pfaffenhofer. Rudy, are you in here? Rudy is not in here. Anyway, Rudy is a professor here at Biosci. And they gave a new idea to find out about molecular approaches to major problems. And they had a whole day for each presentation. And one of them was Chuck Whimpey, who talked on applications of molecular approaches to major problems in aquatic ecology. And they gave unlimited time for discussion, and because there were no big applications yet in molecular biology and aquatic science, he got out a bunch of instruments and started a music symposium of his own. Now, one of his first undergraduate students was during the first year of the RU program, and Trudy Hartman was an undergraduate that worked with angiosperms with Chuck. And you'll see Chuck right there in the group photo, and there I blew him up. I'm not sure who that guy is. And that was when he was a little bit different, but still the same, really. And that comes down to the fact that after all these years, even now, students say, awesome. One of the greatest profs I've ever had. I love this. I love Chuck with every fiber of my being. Don't you want to have that happen to you as a professor? And so he really gets it. And any guy who's spent 2 or more beers at his home at the many parties that he's had for his students, they know this sign that love in life is not a journey to the grave with the intention of arriving safely in a pretty and well-preserved body, but rather to skin in broadside, thoroughly used up, totally worn out, and proclaiming, wow, what a ride. And Chuck's going to give us a ride today. Chuck Whimpey.
Talk. It's been an overwhelming day. I did not rehearse an introduction to this talk, let alone the talk. I'm so glad you could be here, and I'm so glad I survived this long. The talk is kind of, it's chronological. And I just have skimmed across the surface of so many people who have meant so much to me. And that's really what the talk is about. There's some science in it. Okay, there's some science. But it really is about the experiences I've had and what led me to this. So I can, Nicole, how do I?
It's the other one, yeah, the little one.
Okay. Does this work? Okay. So, what led me to this point? I did my master's degree on this plant. This is pearl millet. If you have a parakeet, you probably recognize pearl millet. Master's degree. I did my PhD on this plant. This is duckweed. You've probably seen it in the ponds around, around here. I did my postdoc on this plant, which, as you recognize, is corn or maize. This was the project I was going to bring here. I was studying RNA processing in maize, and that's the project I was going to bring here. And wouldn't you know it, just before I got here, I went to a conference and found out that I had been scooped by a much bigger and better established lab at Berkeley. So, no competition there. But this opened the door to plants that I'd been curious about, and I had wanted to look into them and study them, and that is, these are parasitic plants. These are plants that don't do any photosynthesis. So they break that rule. What's the first rule of plants? Well, they make their own food. These are parasitic plants. And so this opened a whole chapter of research. And actually, Russell Robel was my first graduate student, and he did his master's thesis on this plant, which is Canophilus. Canophilus americana, if you must know. Russell showed that even though it doesn't do photosynthesis, but it has a chloroplast genome, and it's an active genome. There are genes on it, and they're transcribed. For the sciencey ones among you, it has an active chloroplast genome. And we worked on some of these others. This is called beech drops. Probably a bunch of you wildflower people would know that's beech drops. That's Indian pipe or ghost flower. And this plant here is dodder. So this was the most fortunate misfortune that could have befallen me, because it's like, oh, I don't have to work on corn. I'll work on these things that I find so absolutely fascinating. Well, here's another big project that we got into. These are water lilies. For those of you who don't know what a water lily is, these are water lilies. When I came in 1986, there were two other people hired, and one of them was Don Less. And Don was a real plant systematist and an evolutionist. And we threw in together and decided that we could put our heads together for, and do some interesting things. And so that led to a very productive. I can already tell I'm talking too long. I'm just going to flash through these in 10 seconds. A very productive collaboration. We got an NSF grant. We got a bunch of publications out of it, including one in the Proceedings of the National Academy of Sciences. Well, here's another thing that, another seemingly unfortunate thing that had a silver lining. Don Less, and by the way, I should point out that Denise here not only was my first undergraduate research student, but she was the technician on this project. Okay, but then Don left and went to the University of Connecticut. But the silver lining, we got to hire Sarah Hoot? Can you raise your hand? How could we get so lucky? I mean, Don, he was a good friend of mine, but wow, we got Sarah out of the deal. And she had a long and very productive career, it was through interactions with Don and later significant interactions with Sarah that I learned really the ropes of systematics. I learned about phylogenetic analysis. How do you think about these things? How do you look at these things? And so Sarah has been on my students' committees, and I've been on her students' committees, and she blazed the way in retirement. She's the expert on retirement because she preceded me. And by the way, she's sitting next to Art Brooks. I remember in 1986 when I interviewed for this job, and it was Art Brooks who drove me down Lincoln Memorial Drive to what was then the Center for Great Lakes Studies. And he said, "This is and of course, Art had a long and prosperous career. Well, anyway, okay. Now, my interest in photosynthesis led me to this organism. This is, I'll tell you a secret. I wanted to work on this because it had a long name. Proteroeochromonus malamensis. It's not a plant, okay? It's a chrysophyte algae. These are not plants, but they act like plants. They, in more ways than one, they not only do they do photosynthesis, but Denise did her master's degree on this plant, or this algae, and showed that, like a plant, their chloroplast development is triggered by light. Okay? So even though they're not plants, they act like a plant. Well, this is my first lab group. A bunch of them were in the row up here. This is Stan when he had hair. Denise, Russell, Tracy is out on the West Coast. Lisa is sitting right here. Tammy is up north, so she couldn't come. But this was my first group. Okay, these are the ones. The rest of you, Melissa, Faye, Hannah, the whole rest of you. These are the ones that I refer to, Anne Marie, as. Should I go down the list? All right. Yes. These are the ones, Nicole, that I always say. These are your academic older siblings. Because I kind of, I more than maybe others think of these people as a family. So that was my first lab group, and we have kept in touch the whole time. Let that be a lesson. Those of you who maybe are just starting out, you make these are the people who you make friends with for a lifetime. Okay. All right. So how did I get from here to here? Well, actually, it's because I played the banjo. And here's a picture of me at an early field station picnic. That's Ken Nielsen next to me. Carmen mentioned Ken. That was Carmen's dissertation supervisor. He's also a musician, and so he and I played music together. And because of music, Ken got to know Barb. And
I didn't get to see this before.
Barb has not seen this. Nobody has seen this talk. So Ken hired Barb away from her job. She was doing molecular biology at Universal Foods, and it was, it was interesting enough. But she actually was more interested in academic science, and so did not hesitate to move to Ken's lab. And that is how we know Carmen, and that is how we know Russell, the other Russell. There are two Russells in my life, but yeah, that's how we know them. And so she started doing bioluminescence research, and the rest is history. Okay? So what I wanted to do, this is going to be historical. I wanted to describe how biology has changed over these past 35 years, or 33 years. Whoa, get ahead of myself. I'm old enough without adding some years. Yeah, the way that we do this, at any moment in time, you think that you're at the top of the game and this is the state of the art. Okay? You wait. Yeah, you wait. You'll be giving a talk like this and you'll say, wow, we used to do that? Here's PCR. This changed everything, and my earliest little family here knows how it changed everything. This is a method that came out in the mid-1980s, and man, I knew how to clone genes. Then I was, and by the way, Russell cloned a gene for his master's thesis. But right at that time, this method, the polymerase chain reaction, was invented, and it changed everything. So instead of spending 2 weeks cloning a gene, you spend 2 hours amplifying the gene. You can go have a cup of coffee. And we had a lot of coffee. So here's our first PCR machine. So picture, you have those tubes in there and a timer, and then you move the tubes to the next one, and your timer, and you move the tubes to the next. You can't stop. And so, it's like, I need to run to the restroom. And so you'd get somebody else to, get in here, take over the tubes. Yeah. These are programmable thermal cyclers. I never knew, this was our first one, okay? I never knew how to program that. It was hard to program, but Lisa knew how, Denise knew how. Later on, when we could afford it, we got a grant, and we got this one, which was the state of the art at the time. That was a PerkinElmer 380. It's like top line. It doesn't get better than this. Well, actually, Liz O'Brady, who couldn't be here today, she called me up one day and she said, "There's been a flood in the lab upstairs, and it drenched our thermal cycle." I had 2 of these. It drenched them, and so for about a millisecond, I was worried, and then in the second millisecond, I said, "Oh, insurance." And so we got one of these, and in fact, we got 2 of these because I had 2 of these that got fried, and so EWM replaced them with 2 of these. Oh, they're far better. This is a gradient thermal cycler, and with a heated lid. And so even now, this was 10 years ago. Even now, I have no complaints. These are the cyclers we use every day. Yeah. So now, the technique of PCR and the technique of sequencing actually requires primers, which are little short stretches of DNA. Okay? When we started, primers were expensive. They were about $10 a base. So an average length of 20 nucleotides, you'd spend $200 on a primer. So it better be right. So Steve Forst and I bought one of these. And this drove the cost down to about $2 a base. We were on top of the world. You know? Steve is sitting back there, and he remembers, wow, it doesn't get better than this. And we buy them for 50 cents. And we don't even make them, we buy them for 30 cents a base. They get, you know, sent to us by overnight mail. And so, you know, I always say a primer is as cheap as a fast food lunch. So we buy lots of primers. Well, let's try this one. Let's try that one. You know, we'll just try them out. You know? So, yeah, you know, Taq polymerase, which is the enzyme that is most often used for polymerase chain reaction. It was expensive, and there was only one source of it. You had to buy it from this one company. So our solution was to obtain a plasmid clone that had the Taq polymerase gene in it, and we made our own bootleg Taq for a lot of years. In fact, this was a class project. Those of you who've taken my molecular techniques class, we used to do this in class. And then we would 10, you know, you're used to little bits of enzyme like this. We were making 10 mils, and that was every lab group. We were making over 100 milliliters of Taq polymerase, and most of it went to Sarah's lab. It worked great. You know, her students, you know, she's had a lot of students that were, you know, that used our bathtub Taq. Now Taq is one of the cheapest enzymes you can buy. You know, so we don't make it anymore. Just buy it. And you can buy it as a 2 times master mix. This was a great revelation to me when one of my former students, Erica Holson, she said, "Oh look, we can buy this ready-made. It's a 2 times master mix." And I said, "We're tough. We're molecular biologists. We ain't gonna buy no stinking master mix." She said, "No, really, let's just buy it." And so I did, and we never looked back. So now the evolution of DNA sequencing. This is the state of the art when I arrived at UWM. These are this big, okay? It's a vertical gel apparatus. There's an art to pouring these gels. They're paper thin, okay? And then you radioactively label your DNA and you load it on there. And then you have to read this god-awful thing. That is an autoradiogram, and you read up, you read up the ladder. You read there's a base, and there's a base, and there's a string of bases. We didn't know that we thought we were, again, we thought it couldn't get better. I gotta say, I think the person who set the record in my lab is Lisa Van Eert. She had 2 or 3 of these things running all the time. And imagine, the thousands of nucleotides that she read by eye. She read those things. And then she, proceeded to produce a dissertation that was as thick as a phone book. And I should, for the millennials. It's a term you might not be familiar with. A phone book is a book with everybody's phone number in it. So yeah. How's your eyesight, Lisa? I'm surprised you I'm surprised that didn't make you go blind. Okay, so DNA sequencing has changed. Here's the original one, okay? And then an automated version of this came out. This is a slab gel apparatus. You can go up and visit one of these up on the 4th floor. It's no longer used, but you can go visit that, the lab of Linda Whittingham and Peter Dunn. And this, wow, this is the top of the game. Until a capillary version of it came out, which has way more throughput. And so this is a capillary version of this slab gel version, so much higher throughput. This is an example of an Illumina next generation sequencer, and this will produce in the equivalent amount of time orders of magnitude more than this. Okay? And so now there's a bunch of people, in the room that have worked on this. Jake's dissertation. And Jake, raise your hand. And he's not the only one. I'm just pointing out that Jake, is doing metagenomic sequencing of microbial communities in pitcher plants, and you couldn't do this without this technology. Well, now, this is kind of the ultimate. This is a nanopore sequencer, and it's the size of a harmonica. And I bought one because it's one of these things, those of you, the faculty members among you know that, I had the money and I had to spend it before the money went away. So I bought one of these. This has revolutionized sequencing. This is a picture that I pulled off the internet. You can sequence DNA out in the field. Okay? Long reads. Okay? You can do this out in the field. They've done this on the space station. Or even crazier, you can do this in Peter Dunn's office. Because he's got the computer that'll drive that thing. Speaking of computers, I wanted to show you. That's a picture of me typing my master's thesis on a manual typewriter. And literally back then you would cut and paste. You would cut and paste. Like, I want this paragraph down here. Okay? That's where the term cut and paste comes from. When I was all done with this thing, all taped together, and everything, I handed it over to a professional typist, and she typed it for me on an IBM Selectric typewriter. Wow, that was quite a machine. So anyway. This is like the machine that I typed my dissertation on. This is a first generation IBM PC. Okay? And I was telling some of my students about this last week. I said, I should bring in my first laptop. It's down in the basement somewhere. But this is my first laptop, and this is my latest laptop. And I thought, wow, this is so portable. It's this big, it's this thick. And you can break your arm to carry that across an airport. So so I'm sure glad I got one of these. I should say, along with computer technology, now we have email. And in fact, some of you think that email is passé. That's so old school. But I remember I can't remember when, like 1990. Okay? So Russell had, Russell had finished his master's degree in my lab and had gone off to NC State for his PhD. And so some time went by, and I was wondering how things are going, and he called me up. I think he called me up, and he said, "Why won't you answer your email?" I said, "What email?" I said, "Do I?" He said, "You have an email account. I've been writing to you." So when I finally figured out how to get to my email, and then I had to come up with a password. And when I finally figured that out, there were a whole string of emails from Russell. Telling me how he was doing at NC State. Well, I didn't know. Anyway, here's another thing. This is another way that things have changed. People used to publish DNA sequences on paper. Like you'd have to go to the library. There was no internet. You'd have to go to the library, get the journal, and read the sequence on paper. And now, we have GenBank. NCBI has this wonderful database. It's got, I don't even know, billions and billions and billions and billions of sequences in there. Okay? So, and I, I have my students, my undergrads in my techniques class. I teach them how to do this. They sequence genes, or I should say, we have them sequenced for us because we don't sequence genes ourselves anymore. Who would do that? We send them away, and then they come back in about 48 hours. And so I teach them how to use the database, at NCBI, and find out what their sequences are. And this is always an exciting moment. I never get over this. I think that one of the great pleasures that I have, and I share this with Russell Kuhl. I never got tired of the lab, and so it's always like a thrill to me, like pipette, and to read like sequences, so I always have to resist the temptation to read my students' sequences before they do. Oh. So anyway, yeah. So what did we do with all this? Well, I've already told you about Russell's master's thesis on Panophilus, and I've already told you about Denise's master's thesis on Bacteriomonas. Lisa, that big, thick dissertation that she produced was on bioluminescence. So let me just talk about bioluminescence. There's too much for a 1-hour presentation. In fact, I know what I'm going to go over. When we started, there were only 4 LuxA sequences done. Now, LuxA is a gene that is required to produce light. So bacteria have these what we call Lux genes. Clever play on words. We didn't come up with that. It's such a clever play on words. Lux genes. There were only 4 of them known at the time. These 4. And so Tracy Netto, who I showed you a picture before of, she was our original PCR machine. She designed a couple of primers, forward and reverse, based on these 4 sequences. Lisa then improved on those, so Tracy's primers were here, and Lisa made one out here. Those primers have worked on every bioluminescent bacterium we've ever tried, and I got to tell you, we've tried a lot of them. So this was then. Okay. This is now. This is a phylogenetic analysis that Liz Bolton did. Okay. And I don't even know how to do this. I know how to talk about it. I can tell you what it means, but so Liz and Melissa, they actually know how to do this. And so I salute you. I wish you hadn't graduated and gone away. Anyway, we also mapped, this is a cluster of genes called the Lux operon. And we cloned, mapped, sequenced a whole bunch of these. Jesse did one of these. But we did a whole bunch of them. This is just a smattering of them. Barb did one of these, actually. And so we also figured out how to turn these genes on and off. This was a great victory for us. This is Anne-Marie Wanamaker's work. And so this was, to make a long story short, she gave these things an on-off switch. Okay? We could turn light on, and we could, turn it off if you didn't turn it on. So off is the default state. but she showed that you could turn it on. Now, we actually made 2 of these. We also made one using a LAC promoter. This one uses an Arabinose promoter, but we also made one previous to this using a LAC promoter. And and they both worked. You know, I really, you know, I mean, Anne Marie's thesis was this. She showed that this works on a bunch of different species. Okay, so this actually is Barb's work, and this shows you, you know, off, off, on. Okay, and so yeah, this it's a thing that, you know, that we did, and then Liz Bolton did some of these experiments, and Melissa White did some of these experiments, and this was a great victory. So anyway, now our diversity studies, you know, that big phylogenetic tree that I showed you, that got us into ecology. Now here's the thing. My closest colleagues are ecologists, okay? You would think that they would have had the courtesy to warn me about ecology. Here's the difference between molecular genetics and ecology. Here's molecular genetics. Here's ecology. But, nevertheless, we waded into this field, sometimes literally. Liz O'Grady and Mike Kozlowski risking their lives. They could have been eaten by sharks, sampling the bay. You might wonder, well, why are they using a fish net to sample bacteria? It's because we wanted fish, because bacteria live inside the fish. And it's a long story. Well, actually, it's a short story. So, you know, we also had a research vessel. And so this is Mike and Jesse and Liz. And, you know, I just want to point out who was doing all the paddling. So, by the way, Mike and Jesse are in the front row. They had the only barefooted wedding I've ever been to. It was cool. Yeah. Anyway, think about that, those of you who, you know, along the way, you have to figure out how to fix old equipment. You know, sometimes these things are just so obsolete. You think that they don't, you know, you call the company and maybe the company doesn't even exist anymore. Like, well, we have this model number? What? You know, and so you have to learn how to do this. And these are just some examples. This is, you know, this rotor jumped off this microfuge, and so I went to the hardware store and got a screw and a washer and put it back together, and we still use it. And this is a shaker that my former student Paul Carini fixed, and so yeah. And this is what the inside of a microcentrifuge looks like. I figured, you know, when I took this apart and diagnosed the problem, I just bypassed the magnetic lock. So it doesn't lock. You know, there's a magnetic lock, so you can't open it while it's running. And I see Claire laughing. Your microfuge is new. Mine is old. Remember this. Just take it apart. I think actually Russell bypassed the lock on a big centrifuge. Yeah, we had like a string that would, you know. You have to be resourceful because the warranty runs out on your equipment. All right, so there was a whole episode, a whole decade actually, where we were involved in summer research courses. These are courses that we taught at MBL, and I could spend an hour talking about this, but I'll make a long story short. Barb was the course coordinator, and these are the staff and the students. We did this in 1990 and in 1991, and although she couldn't be here, I just want to point out that person right there is now the Associate Dean for Natural Sciences. That's, yeah. Yeah, so she I saw her earlier today, but she couldn't make it to the talk. But there she is, immortalized in the black and white photo. And that was back when she was a postdoc. Okay? So postdocs grow up and become associate deans. For the rest of the 90s, we ran these molecular techniques workshops, and again, I could talk endlessly about each of these. This was Barb's idea. She went to the dean of the graduate school at the time, which is the research arm of UW, and she said, "We want to run these summer research courses." And I'm paraphrasing, but he said, "No, we can't do that. We're not Madison." She said, "Yes, we can. What are you talking about?" So she insisted, and by the way, with the help of, she was working at the NIHS Center, and with the, Jamila, where are you going? There's a whole chapter of my life that involves Jamila. Okay, I'll talk to you. So Barb, when she was working at the NIHS Center at the time, the director of that center was David Petering, and they exerted the sufficient pressure on the graduate dean to let us do this, and we spent the rest of the 90s. This was an international molecular techniques workshop, so we had people from all over the place. Plus, we had a lot of locals. So I would love to point out all these people because some of them are in the room, but I don't have time. Now, in Carmen's very nice introduction, you might have noticed that I've got a lot of work. Here's the list. So now, I love biology. I love teaching. When you love teaching and you love biology, you teach a lot of courses in biology. Okay? So you don't have to teach this many courses. You just have to teach a couple of them really well. Okay? And that really, I'm trying to be serious now. Being serious. But yeah, there's nothing more important than to enlighten students. That's how I got into this, and I suspect that you could go to every faculty member in the room, and they would tell you about at least one or more professors who inspired them. So you don't have to teach this many. It's just that I love biology. So this is all the courses I taught. Which brings me to the Honors College. This has been such a rewarding experience to me, and I got to tell you the story. The previous director of the Honors College, who I had known for years, he emailed me. By then I knew how to he emailed me and he said, I distinctly remember the first line of his email. He said, "I know you don't have time to do this, but when you consider teaching a course in the Honors College, an honors seminar." And so I thought about it for about, again, a millisecond. And I said, yes. And so that launched me into a whole world of teaching these honors college courses. And this is the list of courses. I liked it so much that I kept coming up with new courses, hoping that they would invite me back. And they did. And so I have taught an honors seminar every semester since that time. Jessica and I were trying to figure out what that time was, and it seems that it was about, what did we decide?
2010-ish?
2010. So I want to put a plug in. I think that BioSci has the best faculty. I hope chemistry isn't listening. Freshwater sciences is listening. Sorry, Ryan. A shameless plug. We got a bunch of Herman and Russell. They taught honors courses. Anyway, this is a shameless plug to the Honors College because you will just try it. Just try it. There's no other place that you can. I just noticed Mike Carvey is here also from Freshwater Sciences. They're everywhere. These people, it's like an invasive species. Honors courses are they're not as easy as you might think, but they're so rewarding. So I would encourage you to give it a try. And you could even take over my courses if you want. And so you might wonder, who do I contact? Well, these two. I think that everybody, in the Honors College, the instructors, the staff, they're all just great. But I just want to call attention to these two people because they're the ones that really make it happen. In Lamb, she's right there. Laura Blaska is right there. Good. Now, I want to talk about the Office of Undergraduate Research. This is the best thing that's happened to UWM in 33 years. Okay? And these are the people that I want to call attention to. Now, Nigel is the director, and Tamara and Kyla, and I've been, for years, Tamara and Kyla were just, I talked to them by email, I hadn't met them, but, so, yeah, they're running, they've been running the show for years. Now, you might wonder why I made this picture so big. This is Jessica Schultz, and she's sitting right there. And the reason I made this so big is that Jessica did her undergraduate research in my lab. She also took my first honors college course, which is called Plagues and Pestilence, Past and Present. So I mostly like the alliteration. So I like that. Oh, now I have to build a course out of that. Jessica was a research student in my lab, and now she is paying it forward because she is right in the thick of it. I don't remember your title over there.
Programs coordinator.
Program coordinator at the Office of Undergraduate Research. I remember when Barb and I met Jessica, she was a server at a little cafe that we used to go to, and she marched up to our table with a pot of hot coffee, and said, "Are you Professor Wimpy?" And I said, "Yeah." And so, to make a long story short, she wanted to do research in my lab. She was armed with a hot cup of I thought, if I don't say yes, she'll pour that hot coffee. So, yeah, I said yes. I said yes, and the rest is history. This picture, by the way, could have been taken yesterday, but it was taken about 10 years ago. So. Now, I also have, I was asked in 1994 to teach a course out at the Oregon Institute of Marine Biology, or OIMB, and which is one of my favorite places on earth. They have a seminar room that overlooks Coos Bay, so you can imagine, the distraction, of looking out over the bay when you're supposed to be focusing on the seminar speaker. But anyway, yeah, these are just some of my favorite organisms. So we, I taught out there for 20 years in the summers, and so these are, people who are part of my life, and they're permanently part of my life. Kevin is right here in the front row. Kevin took every course I teach here, and then when he was a master's student at the University of Alaska, he took, working on bioluminescent bacteria, by the way, he took my course, my marine bioluminescence course at OMB. This is our dear friend Anna Brinkman and her dog Jadzia. She took my very first course that I taught at OIMB, and I always tease her about how when I first met her, she nearly killed my tropical sea anemones. So she was trying to be helpful, and she changed their water with that cold Oregon seawater. Anyway, they lived, but they were doing the anemone version of shivering. So anyway, but yeah, she's been in our life ever since. So this is why I say, keep track of these people. They will. This is our kind of most famous bacterium. This is it's a strain of Photobacterium phosphorium. It's the brightest one I've ever seen. And so Kevin was in that class, and so I credit him with isolating this thing. And my colleague, Hunter O'Reilly, now she's Hunter Cole. You can look her up. She has a website. She's an artist and a geneticist. She used it to do the cover art for Nature Genetics. And I've always said this is the closest I'll ever get to making the cover of Nature Genetics. At least my bacteria did. Let me, don't be scared. Can I make this black? Anybody know how to make this black?
Hit B on the keyboard.
Oh, I'm not stupid. If I turn it off, I'll never get
No,
it back
hit B on the keyboard, Charlie.
Huh?
Hit B on the
B.
keyboard.
That's my sister. She calls me Charlie. The real Charlie is sitting up there. So B.
Just B.
Huh?
Just B. Yeah, B. What are you doing? What's going on there?
B. Oh.
Yeah.
I knew you'd be good for something. I've known you since 1965. Oops, I gave away her age. This is Photobacterium phosphorium. It does this little trick where if I let it, if I don't swirl it, it goes dark. But if I swirl it, it goes light. Okay. Now some of you know why. And the rest of you, it's magic. Yeah, and by the way, I didn't know that these guys did this for me. But notice along the walls, I didn't know about this. This was not my plan. Okay? Those are Petri plates. Those Petri plates are inoculated with this, the most famous microbe that's ever come out of my lab, and so yeah. I could do the rest of the talk,
Just hit any key.
Oh, hit any key?
Hit any key. Yeah, there you go.
Did you see this? I think I put the lights on. All right. So anyway, now music. Music has been a part of my life since I was a little kid. You know, but especially my life here in Milwaukee. Okay. So I have to show you a picture. Let me compose myself. These are my closest music friends. We lost Ann and Will about a year ago, within weeks of each other. It was a great blow to us. But I still have Jessica. She's sitting right up there. There's Jessica. Jessica is a multi-instrumentalist, and I always joke with her. I've been joking with her for 10 years that I don't have the energy to retire because I've never seen a person busier than Jessica Worth. She is running from place to place, and every time we were trying to schedule, a music session, and she'd go, "Oh wait, let me check my calendar," and she either had this rehearsal or that rehearsal or some kind of thing. Anyway, this was such a rewarding part of my life, and so I just wanted to call attention to that. Another couple of my really close friends, music friends, Kathy and Sally are up there. If you could raise your hands. We know them because of this, okay? Because of the music. So just want now. I want to, I'm actually, you wouldn't know it, but I'm almost done. But I want to, talk to my colleagues here for a minute, okay? I'm not kidding. I'm not kidding. You're all smart enough to win a Nobel Prize. We made sure of that when you were hired. But we're not hiring any dummies. Okay? And the tradition continues because we hit the jackpot with our newest faculty member. So, over and over and over, we have hit the jackpot. So, even though you're all smart enough to win a Nobel Prize, don't focus on that because they only get one a year in our field. So, you might get one. You're smart enough to get one. But really, there's something more important. And it's a sure thing. Okay? And what that is the lives you touch and the careers you launch. I am so proud of my former students and my current students because I know they're going to do great things. And so that's what I think is the most important thing we can do because our students are our most important legacy. Okay? So, just wanted to say that. Besides, Nobel Prize, what are you going to wear it around your neck? Go to the grocery store with the Nobel Prize. But you know what's better? What's better is when you hear from your students and they tell you how they're doing years later, and it just is so heartwarming. So I wouldn't trade that for anything. So now in science, we like to make discoveries. That's what it's all about. So I just want to show you some of my greatest discoveries. Now this is going to, I'm not going to leave the slide up for long enough for you to go through it because I just want to impress you with these names. This is everybody who's ever been through my lab, okay? And there are some outside my lab. These are the people that, sometimes I'm on their committee, sometimes they've come through my classes, sometimes, it just, I won't leave this up long enough for you to see, who they all are. But I have a message for my students this is an abbreviated version of what of a series of slides that some of you have seen if you've taken my Bio 150 course, at least in the last few years. So this is a message that I leave my students with. Okay, there's a lot more of these, but here's the ones I wanted to show you. Okay, I'm just going to click through these. Now, behind the scenes, there are people who make everything happen. This is not the full list, but I was scrambling around today taking pictures of people. And they probably wondered, why? These are the people who really make it happen. And I don't mean to leave anyone out, but they are so important to me. Starting with Teresa, and Nancy went to, I won't go through all of them, but I just want to say, Teresa, very close friend of mine, close friend of Barb's. Nancy is now in GeoSci, so I have to go down to the third floor to tease her, for her to tease me. So yeah, but yeah, these are all the people that make it happen. And like I said, this is not the whole list, but these are just the people. You might. That is Jenna Rock. And so yeah, those of you who Jenna Rock was actually Jessica's predecessor, and she then went to Florida to do her master's degree. We've stayed in touch. And this is her floating in our bay. So yeah. So anyway, I could go through each of these people and tell you why they're so important, but just know that these are the people who make the rest of our lives much, much easier. Okay. Now, some random photos. I could spend 6 hours on these. I'm going to click through them really quickly. You won't even get to see who's who on these. But I'm just going to click through these. These are some of my colleagues. Look at this guy here with all that hair. I don't know who he is. There's some vague resemblance to somebody I know now. Yeah, Linda, it hasn't changed. Linda, and Peter, Doug, Julie. I don't have enough pictures of my colleagues. You plan one of these things and you go, oh wait, I don't have pictures of these people. But I could have gone to the website, I guess. Boring. Here's Barb and Linda at that same party. So, and here's some other random pictures. These are people who mean so much to me. Some of these are new. Some of these pictures are an hour old. Some pictures are 10 or 20 years old. There's no chronology here, okay? But these are just so important. And Faye, do you recognize some of these things?
You're so young, wow.
Yeah, so young. Yeah. So yeah, I mean, you might recognize, maybe not. Just know that these people are very important to me. Okay. And I can't believe that I don't have a picture of Ari Brenner. Why do I not have a picture of you? I spent an hour going through my computer. I looked at every picture.
I win.
Well, okay. You'll have to look at Ari in real life. So, yeah, boy, this was a group. I can tell stories about them. I can't resist. Let me go back. I can't resist. This is Joanna. She was a ballet dancer. Her undergrad major was ballet. She came to my lab, and she did some of the most astonishing molecular biology. Wow. So I always say, art and music are actually not at opposite ends of the spectrum. They're right there. Okay? So here's some others. Here's Anne Marie. So I could name all these people. Erica Olson is in Congo right now, working with Doctors Without Borders. So here's Denise and Stan. Okay. I have to say about Erica, she was the one who got me into that, buying that pre-mixed pack polymerase. I am terrified for her. I'm terrified. She's in the Congo, working with Doctors Without Borders. So, but I am. Compose myself. I am so proud of her. Actually, I'm so proud of everybody. I just wanted to point out that Erica is over there. Wow, I'm so proud of her. Well, let's see. Here's some other pictures. Here's Liz O'Grady with her first son, Maggie Davis. This is Brett Baker. I feel like I need to call attention to Brett Baker. He sort of fell into my lap. He was abandoned by his advisor and was left stranded. And Barb said, "Well, why don't you go and talk to Chuck?" Kate, where are you going? So because of Barb, I got one of the best students I've ever had. He is busily shaping up the tree of life. So look for his name on microbiology papers. So anyway, I need to move on. Ira Heimler gets her own slide. Ira Heimler she's again one of our best friends. I got to serve on her dissertation committee. She worked closely with Barb and then became one of our best friends. She is hilarious. So I had to show these pictures. This is her in Baghdad. She joined the army at a very unfortunate time. Wouldn't you know that a year after she joined the army, we went to war with Iraq. So anyway, there she is. She spent 14 months in Baghdad. Here she is in a sandstorm. Anyway, here is my current lab group. And Anna, what did you call this? Like bad Photoshop? Anna was not there the day that we took this picture because she had the poor judgment to go to class. So this is my current lab group, and by the way, I'm so fortunate I got to borrow a grad student because Nicole Jimenez was my last graduate student, and but I got to borrow one this semester, and then it's Brittany Vanderhoof. So yeah, this is my current lab group. You would think I would taper off. I know what time it is, by the way. You would think I would just kind of take it easy, And I said I was going to do that.
We didn't believe you.
Instead, I took on 5 students this semester, and they've all done fabulous work. But talk about running things up to the end. So this is Sena Hamdan, and she cloned 2 operons, and we just got the sequences back last week. Okay, 7 kilobases of DNA. This is Thea Park, and she had a very nice project, and she identified this strain that had 2 Lux A jeans in it. And this was like, a week ago or something. It was like last week. I said, I said, you know what we gotta do? We're gonna have to clone it. And so Thea, who's sitting right there, so she just got the sequences back yesterday. So talk about running things up to the end of the semester. So now we have to blast all those sequences. No pressure. Now, I want to tell, I'm getting close to the end, but I said that a minute ago. So as scientists, of course, we like to make discoveries. Well, I started, this is my greatest discovery. This was the trip where we got that's where we, and Denise was alone. That's the trip where we got the organism that Melissa White did her master's thesis on. Fantastically interesting organism that has a giant horizontal transfer. And I know I'm speaking gibberish to many of you, but they got the gene from elsewhere. So anyway, okay, 2005. There's Barb's, this was a poster she did at ASM meeting. Later on, she graduated. Everybody thought that she had an advanced degree, but she didn't. And she decided it was high time to get one. And so there she is. These are my greatest accomplishments. Monica, sitting right there, surprised me by driving over here from St. Paul today. And the others live on the coasts, one East Coast, one West Coast, and they couldn't be here. But when I arrived at UWM, these 3 were the ages of these guys. Okay. So, my greatest accomplishment. So what's next? This could happen but in all seriousness, here's what I plan to do. I plan to do all these things. I plan to write, I plan to lecture, I plan to do public outreach, volunteer. Okay. There's some other things I intend to do. Play more music. I think I'm the only person, well, I shouldn't say that, I'm the only person I know who held auditions for the job of son-in-law. These are 2 of my 3 sons-in-law. The third one is a tech wizard, so I thought, oh, okay, that'll be useful. Play more music. Barb got me a really nice camera. Did I bring it down? Oh, I must have brought it down. A really nice camera. She said, here's the deal. I'll make you a deal. You have to take as many pictures of grandkids as you do birds. So I tried to honor that agreement. So I take a lot of pictures of birds. I had to ask Peter Dunn what these birds are. And because my name's furthermore big tall bird. What sort of bird? A white bird with a curved beak. So another thing we're going to play with the dog is taking a liking to floating in the bay with Barb. Here's my grandson Silas walking the dog on the seawall. I want to teach our grandkids to sail on our bay.